All right, we can get started for today. I will say this is a day that I've been quite excited, but also intimidated for because bioreactors are not my field of expertise. I am a biology major, not a bioengineer. So we're all learning together here. But for our agenda today, we'll talk for 15 minutes or so about the reading discussion and then go into the content for today about what attributes you want to think about when designing a bioreactor, what types of bioreactors are out there, what current companies are working on, what scale some companies are working on, and then we'll start working on your final project again. So I hope you all had a chance to read the paper from last time on decellularized plant scaffolds. So I know this is a scientific paper and it might be confusing to some MBAs or people who don't know a lot about biology or material science. So I wanted to keep it general. First, what do you think is novel about this technology compared to other bioscaffolds, or what is the benefit of using this type of bioscaffold compared to other scaffolds that we thought about in Jasmine's class last session?
I think one really big advantage of using plant tissue scaffolds is that it leaves the vascular network in place. So that way, all the cells can get oxygenated and stuff. So that's a pretty big advantage.
Yeah, that was one of the two big advantages that I was thinking of. That in the paper they were saying how it's actually quite hard to design a vascularization inside scaffolds. So to have these leaves that already have a vascularized system and then you're just decellularizing it is in a way probably easier to do than trying to bioprint a scaffold or 3D print a scaffold. Yeah, so then there's like a smaller benefit to leaves in addition to the vascularization. Compared to other materials, how common are leaves in our daily lives?
Very common.
Yeah. And they're pretty cheap too, right? Scrape them off the ground, have a whole bucket full from raking in leaves. So it's pretty important that they're very readily available and very inexpensive. So they could be quite useful compared to these very expensive polymers that we have to make in the lab. Cool. So then the second question that's sort of more in depth but goes into more of the paper is what experiments are important to perform on bioscaffolds, or another way to think about this is what attributes are we looking for in scaffolds that you saw in the paper?
I think I remember this from the paper, but I don't know if this was like just part of my independent research, but I think in the paper they like did some stretch tests, essentially, like just making sure that the scaffolds were like flexible enough to sort of like move around. Like if this was being used for tissue engineering, then the graft like wouldn't break or anything like that.
Yeah, exactly. So we talked last time that the mechanical properties of the scaffold are quite important for the cell viability and cell behavior. And even that topological cues or geometric cues from the scaffold can partially replace chemical cues that are more expensive to add in. So mechanical properties were a big test. And if you want, what might be easy is if you like pull up the paper and just look at the subsection titles to answer this question.
Hi. So actually, I didn't have a time to lead article, and also I was not able to make it last time a scaffold session. But I think in general, safety and the biocompatibility of scaffold will be important, I guess, and also edible or not could be to make a steak or something. Yeah. I don't know.
Absolutely. You want them to be biocompatible, both in terms of are they non-toxic for the cells themselves? Do you need to add a coating to make them adhere to the cells? As well as if you are keeping the scaffolds in the final product, are they edible for our bodies? Can they be digested, degraded? Hi, Fernando. We're talking about we're going over the reading from last time. So do you have any factors you want to think about for question 2? Like what properties are you looking for in scaffolds?
Hi. Yeah. Okay. So, a scaffold should, for tissue engineering, should mimic, as closely as possible the in vivo microenvironment of the cells that we are trying to grow on it. And some important parameters to assess are, for example, the stiffness and the elastic module of the scaffold. For example, I don't know exactly the experiments to perform in order to
yeah, so they talked about mechanical testing as one part of that. But then another part of that is the perfusion flow. How well does the vascularization actually work to allow the perfusion of fluid like blood or oxygen and nutrients to those cells? Yeah, so the other things that I was thinking about was cell adherence, the vascularization density and pattern, and whether that's maintained during the decellularization process, like how much is disrupted by that process. And then you also want to think about the decellularization efficiency. So how well has your scaffold actually been decellularized, and how they tested this in the paper was the DNA or protein quantity that was left over from after the decellularization process, because DNA is from cells. And they found that actually I believe that the decellularization wasn't completely efficient until they used it, and unless they used a detergent. And it's quite important that these scaffolds be decellularized, 1, so that there's room for the new cells that you want, but 2, so that you don't cause an immune response from these new muscle cells or whatever cells that you're growing. In this paper, they were actually growing cardiomyocyte cells. Cool. So that's a reading discussion. Yep.
I have one question. Already like optimal, with regard to mechanical properties of scaffold, already like optimal functions or optimal property are well known for culture meat, or it is still under research stage? I mean, like a stiffness, or there is something optimal stiffness is already well known. I mean, to grow fast or to grow mature of a meat.
Yeah, I'm not sure exactly all the science, but I think that it's we know at this point what is the ideal stiffness for different cell types, or at least we know what is the environmental stiffness around each cell type in vivo. Yeah. And it's actually different for muscle versus fat. So you have to deal with those different needs from their environment when you're creating scaffolds.
I see. Thank you. So this kind of how can I say, the paper or reading, we can find such kind of number or values.
Yeah.
Okay.
Yeah.
Thanks.
I don't have them readily on me, but I think you can Google search them.
Thank you.
Okay, cool. So are there any other questions about the reading? Hi, Meng. Okay, if there's no questions about the reading, we'll go on to bioreactors. So Fernando might know more about this than I do, or at least from, well, he might be able to teach this class better than I can in a year or so.
Okay.
Because Marian Ellis is fantastic, one of the leaders in the field in this particular area of bioreactors and overall bioprocess. But when we think about bioreactors, the main definition, the main definition for bioreactor is a automated controlled chamber for cell growth, or I would say not just cell growth, but cell life, because you at some point don't just want them to grow, like proliferate, you want them to differentiate or make a certain biomolecule that you like, for in the case of precision fermentation. So the main attributes you want to think about in making a bioreactor is you want to maximize the time that you're in exponential cell growth in the case of proliferation. So you want those cells growing at their max rate. You want to have minimum space requirements. So this would be maximizing the cell density that you can have, and you can maximize that cell density depending on the type of bioreactor that you're using. So Marian Ellis is especially working on this variable where she's using hollow fiber reactors, because these are some of the best bioreactors for maximizing cell density. So you also want to think about having a minimum energy requirement, because bioreactors are the second largest component of the greenhouse gas emissions, and definitely the energy use for cell ag. Can anyone remind me what is the first largest component of greenhouse gas emission for cell ag?
And the media production. Okay.
Yes, media. And even media is like partly related to the bioreactors, because part of the reasons why media is so expensive is because it requires these growth factors, and we grow these growth factors in bioreactors. So we also want to have minimal consumables, or at least have a good efficiency of the biomass that we're putting in to give the cells, and how much product we're getting out. And we want to have the process of bioreactors be as automated as possible. The whole reason that we started designing bioreactors in the first place was that it's incredibly inefficient to grow tissue cultures by hand. Like 12 hours a day, your life is completely consumed by cells. If you've ever done biology research before, I know I have, and it's such a pain in the butt to have your life in a lab revolving around the cell cycle. So the reason that we have bioreactors in the first place is to have it automated, both for labor costs, and then also whenever you are touching those cells or having manual labor, there is more concern about contamination. So you want it to be quite contained, automated. So the first one we said there was exponential growth. Exponential growth of cells occurs when the cells are unlimited by resources. So they have enough resources both in the bioreactor, but also those resources can reach your cells. We also want to think about, we said, having minimal consumables, or having a good conversion ratio of the biomass that we are producing, or the products that we're producing, versus the amount of media we are consuming. So you might measure this term called a yield efficient based on how much glucose you're consuming, how much nitrogen, how much growth factor. So one of the things that's really important when designing bioreactors is this idea of scaling up. You start making a bioreactor. There are smaller bioreactors that are more lab-sized, but you tend to go to a bioreactor when you're looking at scaling up the volume. So when you are scaling it, you have to have a lot of thought to how the environment will change with this scale. So you need to, so one of the things that you need to think about is how well you can have biomass transfer. And one way is to improve your biomass transfer, like how well those resources can get to your cells, is to improve the speed of flow of materials around your cells. But then you also have to balance that with the idea of the shear stress that you're putting on those cells when you're exposing them to a fast flowing fluid. And then we said to have a minimum energy requirement is quite important. And this will change, depending on where in the world you are. So as also from Ellis and as well as Tuomisto, there have been a number of life cycle analysis reports about that have found that your energy requirements in your bioreactor will depend on the ambient temperature. So it will require more energy to grow your meat or your bacteria or yeast in Wales, for instance, than it will in Kenya. As well as you have to think about what is available in your environment. So is there, is fresh water difficult to come by in your particular area, or can you use different biomass depending on where you are? So like you can use cyanobacteria, wheat, corn, depending on what ingredients are available for you. Okay, so feel free to stop me at any point with questions. But now I'm going to talk about the different types of bioreactors there are, and there are a couple of different ways that you can categorize bioreactors that can make them confusing. So the first way you can categorize them is how you're feeding the cells, or what flow of material there are around cells. So the easiest way to have flow of materials is just have a batch fermentation, where you put in a given amount of media, you put in a given amount of starter cells, and grow that until the batch is ready. You can also have a fed batch system where you have a flow of your biomass or media into that fermentation tank. Or in a later design, you have what's called a continuous or perfusion system, where you are constantly flowing new media over the cells. You have a system to capture the cells, but otherwise you have a way to also allow that media to flow out of the cells. So you're constantly washing the cells with new media. So then you can also think about this as different ways that you are mixing the media or replenishing the media in the cells. So you can have agitated bioreactors, where you're having a time averaged homogeneous environment using different types of flow. Or, and this can be batch or fed batch, or with different added features, you can make it continuous. Or you can have a perfusion system, like we said earlier, where you have a steady flow of media over a cell population. So a stirred tank bioreactor is one of the most common types of agitated bioreactor, while a perfusion bioreactor in its most simplest form, I think, would look something like this. So Fernando, you're probably going to work with Alan, Scott Allen, in your research, but he wrote a quite influential paper in this topic on an agitated type of bioreactor called the stirred bioreactor, as you said before, where you have, I think it's the easiest one to wrap your head around, where you have this mechanical stirring that allows for this, the media to circulate around the environment. However, this is more of a time averaged approach. It can lead to necrotic centers because your cells get cut off from nutrients. And it also has it can put stress on your cells, and cause them to disassociate from the scaffolds. So this is a sort of bioreactor that's most useful for non-adherent cell types, which, as we said, I guess 2 weeks ago now, muscle is not a non-adherent cell type, or it needs to adhere to a scaffold in order to grow. So another type of bioreactor is that the Alice Lab is working on is the hollow fiber bioreactor. And this is, I think, one of the most promising bioreactors out there, although there's not really one bioreactor type that has been ruled out for cultured meat or for precision fermentation. But hollow fiber bioreactors, you can see how they work in this diagram here, but basically there's a given number of these porous straws that act as scaffolds, and your cells adhere to these, and they have media that flows within the straws and then perfuses out of the pores in the straws out to the cell environment. So this bioreactor is quite intriguing to me because it has some of the highest possible cell density compared to different types of bioreactors, and also it's easily scalable in that these straws or the fibers make it easier for the cells to act like they are not at a huge scale. They create a more closer knit environment where they can really feel that those nutrients on them. But the other types of bioreactors there are things like packed beds or fluidized beds, rotating walls, wave rafting beds, spinner flasks, airless. And these are basically different ways that you move the bioreactor around to get a flow of media over the cells and remove metabolites. So in addition to those different ways that you move the media or move the fluid around the cells, there are different bioreactors considerations that you want to think about depending on whether you are aiming for cell proliferation or differentiation. So when you're starting from when you start from your initial biopsy, you can use what's called a seed train to sort of help train the cells to grow into a larger bioreactor. And then you also want to think about additional features to that large tank. You have to think about the media storage tanks that you saw in a fed batch or continuous process, you want to think about ways that you can regulate the heat that's in that system, as well as have sensors for pH, temperature, and this, I forget what the name of the lab that's doing this is, but I can link it earlier, but there's a whole lab that's just working on different sensors for bioreactors. And yeah, making sure that you have a common constant and homogeneous temperature and pH environment throughout the bioreactor. So a different way that you can categorize cell types is whether you're thinking about a single use or a stainless steel bioreactor, reusable bioreactor. So single use is cheaper at smaller scales. It means that you don't have to clean out your bioreactor between uses. So it allows for faster production time. This might be useful for a smaller production system like a DIY home system or a local community bioreactor. But eventually, when you think about scale, stainless steel is less expensive. So this is what a single-use bioreactor looks like. You have these bags here, and you can change those out after you have your batch. So single-use, in some ways, it's more environmentally positive because it has less water and energy use because you don't have to clean it out between batches, but it can be more expensive long-term because you have to buy a new bioreactor every time, and it is going to leave a lot of that plastic waste that we see. But it does allow for a faster turnaround time than stainless steel. But so far, the cultured meat companies that I've seen, I don't think any of them are using single-use bioreactors, and instead they're going straight to stainless steel. So the three cultured meat companies that have already started working on pilot plants where this large bioreactor at scale process is Supermeat, Just, and Biotech Foods. As well as there are companies like Culture Biosciences that can sort of rent out their ability to run a bioreactor system for smaller startups, so that those startups can check to make sure that their system is working not just at a lab scale, but can also work at these larger scales. So these are the current players of bioreactors. So Ellis's group is called Sire Agriculture Limited, I believe. But Fork and Good is looking at a cool system that we talked about earlier that uses different cell types instead of media, as well as inter-agriculture. You have bioengineering giants like Merck and Thermo Fisher that are also working on these technologies, as well as Future Meat Technologies that's working on how to optimize the system for smaller scale cultures. So they're using, for instance, media recycling to make it more efficient to even just have a smaller scale culture, whereas a lot of times you're trying to maximize the amount of scale. So I know that I don't have a lot to say on this topic, so I have a ton of resources that I would recommend that you watch instead of talking to me, because this isn't my area. But hopefully you can learn some from 1037 if you're at MIT, or learn from Alice's number of talks, as well as this resource from Elliot Schwartz. It's quite useful for Bioreactors 101. Okay, so that's the end for bioreactors. Does anyone have any questions?
Yeah, hi. I have a couple of questions. Sorry, probably I missed, but with regard to hollow fibrillary membrane, what will be the key differentiation point or advantage compared to agitated current system? What is the main difference or advantage? High cell density.
Yeah, so I guess the high cell density is one of the largest advantages, and as well as that it's easier for the cells, it's easier to adapt the cells to this larger scale to a larger scale environment if you use this type of bioreactor compared to other bioreactors that are more difficult for the cells to feel like they are safe at the scale.
I see. And then this membrane is not commercially available. It's currently like a research phase, or already this is already commercially available?
I am betting that there's hollow fiber membranes that are already commercially available, because bioreactors are already like this are already used for a number of fields outside of cell ag.
Mm-hmm.
Yeah.
Do you know what kind of material they are using for this membrane?
That I do not know.
Yeah, it's okay. Yeah, thank you. Yeah, I see. And what kind of other applications for this membrane? Sorry, very basic question though.
Sangeeta, what other fields use bioreactors?
I think in general, fermentation is fermentation is kind of where this bioreactor development was first pioneered, I think. So a lot of, anytime you need to culture a lot of microorganisms or react a lot of chemicals at once is one place. I think a lot of, chemical engineering in general, this will be used for a lot of kind of large quantity reactors.
I see.
Think of any other fields outside of that might use it. For example, ethanol production is a huge one that can happen in bioreactors. But
yeah.
Thank you so much. Yeah.
Okay, so I found this earlier and wanted to just put it here that I think next week or about 10 days from now, there's going to be the Precision Fermentation Innovation Summit. So it's there a lot of the events that I've seen on cellular agriculture are focused on cultured meat. So if you want to learn more about precision fermentation, I would strongly check this out. Tickets are not free, but there is a discount here